All right. Yeah. Thank you for joining today's One World Signal Processing Seminar. Today, I'm happy to have my good friend, Professor Joakim Jalden from KTH of Sweden as our VIP speaker. We know each other for almost 20 years, I guess. Almost. Well, not yet, but
Yes.
almost. Yeah. And then I can really tell you that he's some of the most talented and most well-educated researchers I have known for our generations. And yeah, there can be so many things I can talk about Joachim, but I have to try to be concise. Well, since this is a short introduction, now his research is most famous for MIMO communications. In particular, if we talk about MIMO detection. Now, his research in MIMO detection is really powerful. It's not only about building algorithms. In fact, when he built algorithms, those are some of the very amazing and powerful algorithms. His research is very powerful when you talk about deep mathematical understanding of MIMO detection algorithms. In particular, if you talk about a class of branch and bound method known in the literature as phi decoding. Now, in fact, I myself also work on MIMO detection. There's one thing I can tell you. There are always something I cannot beat Joachim over the course of the past 20 years when you talk about MIMO detection research. Now, he's currently shifting his research interest to biomedical problems such as cell tracking and cell detection. In fact, today's talk is about cell detection and how sparsity could help you do a reliable counting of cells. Now, here is a very quick bow with Joachim. He received MSc and PhD at KTH of Sweden. He has been with Stanford, Vienna University of Technology, ETH as Zurich, Switzerland, either as a visiting student, as a visiting researcher, or as a postdoc. He received IEEE Signal Processing Young Author Best Paper Award because of his research on speed decoding for MIMO detection, and also another career award pronounced as Ingvar Carlson Career Award which is by Swedish Foundation of Strategic Research. He also got several awards for his latest research on cell tracking in ISBI, and he is currently signal processing for communications and networking technical committee chair. So without further delay, I will pass it to Joachim, and let's welcome him.
All right. Thank you very much for the kind introduction, Ken. So soon, in 2003, it'll be 20 years. 2023, it'll be 20 years. It started actually with ICASP in Hong Kong during the first SARS outbreak, which is, I guess, a good connection to these times. That's when we first met virtually at that time.
Yeah, the very unfortunate thing is that year, well, Hong Kong was hit very hard, yes. And I would be able in Italy by June. But right now, we are not knowing when we can travel.
Right.
Anyway.
Right. So let me start with a short background. So as Ken said, I come from a background of wireless communications. I got an opportunity in around 2011 to work with tracking stem cells in time-lapse microscopy. And being a communication engineer, of course, we solved this with the Viterbi algorithm. But that work came to the attention of a company that approached us in 2015 with these images of spots that you can see up here. And they asked us if we could come up with ways to count the spots, which was very important for their particular application. And that took us on a journey through optimization and trying to solve this problem in the best possible way. And it actually went quite rapidly. So already in 2018, around 3.5 years after we started this research, or started talking about the research, they actually had a product on market that implemented the type of algorithms that we developed during this project. So before going into detail, let me give some acknowledgements. Of course, the work is done by PhD students, so in particular, Paul de Guillapla and Claes Magnusson. So Paul is the one that really dug into the optimization within this project. Claes was the student that did the cell tracking research and also helped out in the beginning of this particular research. And then special thanks, well, especially to Mabtech, which is a Swedish biotech company, which is the company that approached us to solve this problem. Also to Quamcom, which is a consultancy company with high expertise in signal processing. These are the people that help develop the hardware and production level software in order to make a product out of this. And then to the Swedish Research Council as well, which has funded part of the research. Right, so in order to bring you to the same page as everyone else on immunoassays, I assume a large portion of the audience are not familiar yet with immunoassays. So let me give you a crash course before coming into the signal processing and the mathematics behind this. So you have to imagine that you're a researcher in the lab, say that you're developing a vaccine for COVID-19, perhaps. What you may do in that case is to isolate cells from the immune response and cultivate them in a plate with microwells. And then you have something to test. It could be a virus particle. So you may add that to one of the wells and see what happens. So if you look inside, deep inside of one of these wells, something like this would be the case. So you have your cells from the immune response in the liquid. You add your virus particle or the vaccine that you're testing, and a certain number of cells get activated. And when a cell gets activated, it starts producing small protein molecules called cytokines, which diffuse in the liquid medium. And some of these are captured by antibodies on a substrate below the cells. Right? And after a while has gone, you wash away the cells, and then you fluorescently mark the particles, and this is how you get a picture showing where those particles were captured in the assay. Right? You have 96 wells, so in one of these plates. So you can do simultaneous measurements of several different cases, and typically you want control measurements and so on in this. If you're interested in the size of this, one of the wells is roughly 7 millimeters wide. So it's not about microscopy. So you take an image at pretty much 1-to-1 magnification ratio onto the image sensor of the camera that you use. So if you take one of these wells after having incubated cells, tagged your captured cytokines with fluorescence, and you take a picture straight down the well, what you will get is a picture of this type, right? So here we're looking down into the well to these captured cytokines, or actually at the fluorescence tagged to those captured cytokines. And you can see the well edge here, right? But this is the circular area that we're interested in. So how many cells were activated by the vaccine or the virus particle that you were interested in testing in this particular experiment? And just to give you a sense of scale, so a spot in one of these pictures is roughly 200 micrometers. So small, but not super small, not atomic level small at least. And the very nice thing about these fluorospot assays, which is a development of a previous technology called ELISpot, is that you can do this at several different wavelengths. So since you excite the fluorophores with a certain wavelength, you can excite several different fluorophores and capture the different light at different wavelengths. So therefore, you can do multiple parallel experiments per well as well, right? And then if you superimpose these two fluorescent pictures, so the green, which is a fluorescent dye called FITS, and the red, which one is a fluorescent dye called Psi3. If you combine red and green, you will get yellow. So if you see in the combined image, you will have some green spots, some red spots, and also some yellow spots, right? And where you have a yellow spot, that means typically that you have a cell that produces both type of proteins that you image in these two experiments. Right, so you can distinguish cells in several different classes, producing one or two, one or the other of those basic proteins, or producing both. And of course, you could have an overlay by random chance, but you can work this out by just looking at the statistics of the probability that you'll have two spots in exactly the same case, and make a good case for you actually having multi-producing cells. So fluorospot is what's called a multiplex assay for this reason. But also for this reason, in the image analysis, you want to locate and count the number of spots, but you want to count the number of spots, but you also want to locate them precisely, so you can compare the location of the spots in order to do this inference if you have multi-producing spots. So at the end of the day, you want to produce something that does something like what you see in this picture, right? So if you zoom into the picture, you'll have these spots, and you want to label them. You say, what type of spot is this? How large is it? Where is it? And some data relating to each spot. This is a particular experiment with something called INF gamma, and I included the Wikipedia entry for those of you who are not biologists here. But for those of you who are into signal processing, the thing to note in this picture is that you see that you have a number of spots here where the intensity has been cranked up so that you can see them, but you also have 3 spots lying here. So the dynamic range of the type of images that you get is very large. So the difference in the intensity between a strong spot and a weak spot are many orders of magnitude. And this makes the problem more challenging from a signal processing view when you both want to detect these spots as well as these spots in the same assay. So to capture why is this problem challenging from a signal processing point of view? Well, if the spots looked like they do in this picture, it would not be challenging, right? So if they had the same intensity and the same shape, you could simply do a template matching or a matched filter to pick out the position of the spots. But this is not what's happening in reality. So in reality, it looks something closer to this. So you'll have spots of different size and different intensity, and strong spots may occlude weaker spots as well, which makes it a bit challenging. So digging into this example by drawing a line through the 4 spots that I conveniently placed along the line here, you see the intensity curve here, right? And there are a number of ways, standard ways of addressing spot counting problems. So the first and the perhaps easiest would simply be to apply a threshold, right? And to threshold anything where intensity goes above this threshold. The problem with that, of course, is that if you have several spots lying close to each other, you may segment those as a single group, and you have to come up with some heuristic in order to break up these clusters. And you can play with the intensity, of course, in order to try to break up the cluster, but that may lead to you missing weak spots or spots of low intensity, even if they're large in this case. And in this case, you can see, even though you can break this cluster into 2 spots, there is no level that can identify this spot here, which is partially occluded by the strong spot. Right. Another possible approach would be simply to look at the local maxima, and in this particular image, that would do actually quite well. You will get 5 out of the 6 spots, but you will still lose this spot, which has been occluded by the strong spot next to it. So you can clearly do better than this. And also about counting local maxima. Of course, your data will look something more like this in reality. You will have noise on the data, which means that you have to do some form of denoising before you pick out the local maxima, and that will further reduce the resolution of spots or the number of spots that you can possibly resolve in these experiments. So we looked at the problems. So the problems, just to reiterate them again, so they have varying size, the spots, they have varying shape, and they have varying intensity. And of course, you have to deal with image noise as in every application. And in particular, a limiting factor here is that strong spots partially occlude the weaker spots. So if you're working with high concentrations, you have to have a more clever approach in order to address this. And what we did was to develop a model-based source point localization algorithm. So we developed a model for how the spots form, relying on the physics of the underlying assay. And this is why I showed you the introduction. I'll go back into that as well to explain the physics model. And then we posted as an inverse problem for source localization and applied optimization algorithms, which I think will be familiar to a large portion of the audience for this particular presentation. So let's look first at how spots form. So we saw the previous high-level image, but let's go back and look at the single cell. So the line that goes across the image here is supposed to illustrate the membrane. The half circle here is representing a cell, and the blue dot is a particle or a cytokine, which gets released by the cell. Right, and as it released, it starts diffusing in the liquid medium up above this membrane. It moves around in some random fashion in a Brownian motion. At some point, it may get stuck to the membrane, and what really happens then is that it's caught by an antibody which this membrane has been coated with. But the membranes are not perfect, so a particle may break free from the membrane and start moving again, right? And it may get recaptured again. Now, a cell doesn't release a single particle, but it releases many particles. So the situation will look something closer to this, where some of the particles will be in diffuse motion in the liquid, some of them will be stuck to the membrane, like this. And at some point, you wash away the cells and the free-moving particles and the liquid. You're left with the particles or the cytokines that are captured by the antibodies. So across the surface, some portion of the antibodies will be holding on to a cytokine. Then to each one of these cytokines, you attach the fluorophore. You excite it with some light of a certain wavelength, and you get the fluorescent response. And this allows you to take the picture, which allows you to essentially image the density of bound particles across this membrane. And this is how the data is formed. And from this example, we can see a couple of things. So obviously, the shape of a particular spot is determined by how many particles were released from that particular cell that was the cause of the spot. But it's also important of when those particles were released. So if particles are released early during the incubation, they will have longer time to diffuse away from the cells. So if you incubate for too long, you'll get very diffuse spots. Or if a cell is turned on late in the experiment, it will typically be weak, but more distinct in the picture. But this will cause different shapes. And then there is a number of factors which on a global scale affect the spot shape. So the diffusivity of the liquid medium, for instance, the capture affinity of the antibodies, so how good are the antibodies at capturing a cytokine and holding on to it. The opposite of that is the dissociation probability. So how likely is it that an antibody once, or a cytokine once captured by antibody, breaks free and starts moving in the diffusion again. And then, of course, for the imaging pathway, the strength of the fluorescence, the optics of the camera, and the exposure time and things like that also affect the image that you actually get, right? But the first couple of reasons are the things which actually change the density on the membrane. So we wanted to have an observation model for this to be able to describe what happened in these experiments. So there are a number of quantities which are relevant to build such an observation model. So the thing we are ultimately interested in is the density of bound particles across the membrane. So this is a two-dimensional entity that's also dependent on time. So it becomes a three-dimensional entity, right? So somewhere on the membrane, there are particles of a certain density bound or at a certain time. And particular to these experiments is that you cannot observe this over time. So you can only decide when you want to observe this, which is when you wash away the cells and fluorescently tag them. So you only get a single snapshot in time, right? So your observation will be a two-dimensional object of this three-dimensional object at a particular time. But then as an intermediate quantity, you also have the density of particles which are in free motion in the liquid medium. And this is a three-dimensional, has three spatial dimensions in and a half plane. So everything above the medium. And the cell at the scale of where things happen here, the membrane is so large you can view it as an infinite plane when modeling this. But it also depends on time, of course. And then very importantly, you have a source density rate, the rate at which particles are introduced into the mix. And they're introduced by cells which reside on the membrane typically. So they have two spatial coordinates, and the production of cytokines over time may vary for cells. And since these are cells isolated at a particular spatial position, you would expect that this source density would be spatially sparse, but not necessarily temporally sparse. And then in order to get the model, we simply link those quantities. So there is a number of partial differential equations which govern this, or one partial differential equations and boundary conditions. So the first one is probably familiar from physics at some point during the education. So this is simply the diffusion equation, which governs the movement of particles in 3 dimensions. So the free particles. Then the next one is also quite straightforward. So the rate of change of bound particles. So the particles which bind to antibodies are the particles which are close to the membrane. So it depends on the concentration of particles at the membrane times some proportionality constant, which is the capture affinity of the antibodies. And then since the particles may break free, you have a term which affects the rate, which is proportional to the density of already bound particles with some proportionality constant, which relates to the dissociation probability. And then the third equation, what that one says is that the flow of particles into the liquid medium is exactly the number of particles that you release from the sources, the cells, and then plus the number of particles which break free. So since this is the increase in number of bound particles, it becomes minus the derivative with respect to time of the number of bound particles at that particular position. So these three equations govern or describe the physics of the assay, or what's happening here. And similar sets of equations have been solved in physics before. It very much depends on the form that you give the solution in order to make it workable in this particular application. So we spent quite a significant amount of time to come up with good parameterization of the solution space to these equations as well in order to build the observation model. At the end of the day, the observation model looks according to this. So you have the observations, and the observations are modeled as Gaussian convolutions of a quantity which we call A here, and then integrated over a sigma. So the fact that you get Gaussian convolution with Gaussian kernels should not be surprising if you know how to solve diffusion equations. So the green function of the diffusion equation is a Gaussian in the number of dimensions that you have in the diffusion. And here we only look at spatial positions. We only consider movement that is in line with the plane where they are captured. And you can write this in the following form as well. So you have Gaussian convolutions of different standard deviations applied to a quantity that changes with the standard deviation. And this quantity A, which is the same one as here, can be obtained from the source density rate by the following equation. And I said before that depending on when you release a particle, it has different amount of time to move. But you cannot just look at the time since the particle was released, because a particle could, with some probability, have been captured for a certain portion of that time. And that's captured by this phi function here. So this one essentially says that a particle released at some particular time ago, what is the probability that it's been in free motion for a given amount of time? And then since it's more convenient working with diffusion distance than time, we do a reparameterizations. We describe everything in terms of a diffusion distance, right? So what this equation is saying is that all the particles which are conditioned at a certain diffusion distance, well, what you expect as the response of that, or the impulse response, should be a Gaussian with the same standard deviation. And then you sum up all of those contributions. And it's quite interesting, actually, that you can actually work out what this probability is if you want to. And that's also something that we did. It's a little bit messy here. So there were previous in the literature, people had worked out what's it's as a time from the release of a particle until it's captured the first time. And that's given by this equation here. The way to do this is to move into the Laplace transform domain over the spatial and temporal dimensions and solve it there, and then move back. Not trivial, but straightforward with tools. And if you look at this whole equation here, so the J, which is in the sum, conditions on the number of times that a particle is captured. Right? So if you only have J equal to 1, you only have this other phi term here, which is this movement distance. But if J is equal to 2, it means that you had a particle which moved, was captured, released, and moved again. And since the process is memoryless, what shows up is simply the convolution between those two subtimes to give you the full time. Right? That's why you get the convolution. But if a particle should be in movement, captured, and then released again, and then the movement for a certain time, then for the time when it's bound, it has to release at least or exactly one time. And the probability of that happening is given by the Poisson distribution with a certain intensity that depends on the amount of time it was captured and proportional to the disassociation constant. That's why the Poisson distribution works up here. And then you simply sum over all possibilities of being captured there, and you get this quantity. And this is not so important for the actual solution, but it's nice to know that you can actually work this out in closed form. So you have the observation model here. It builds on Brownian motion in X, Y, and Z. The absorption and desorption regulate the Z movement and these probabilities, but not the Gaussians themselves. The X and Y motion depends, as I said, on the total amount of time in Brownian motion, according to the green function for the diffusion equation. And then you have this quantity which summarizes the effects of the absorption and the desorption and capture that in the free time of motion. And we make a change of variable simply because it's more convenient. So this mapping here from the source density to this quantity A, the thing to note here is that it only acts on spatial coordinates. So very importantly, this one will keep the same spatial sparsity structure that we had in S, which is important. Since this is probability, it's positive. The source density rate, so cells can only release these particles, they don't absorb them in our model. So this is positive, and that positivity is retained by the quantity A. And also the mapping from S to A depends on the diffusion constant of the medium, the capture affinity, and the dissociation constant. But the very nice thing is that once you have A, nothing more depends on these constants, and they are quite hard to estimate. So it's more convenient to work actually with A than with the source, because then you don't explicitly have to know these quantities in this particular setup. So what we propose as a solution is to recover this quantity A, which we call the post-absorption desorption source density rate. So you think of it as a source density rate, but it's modified in terms of the temporal content to this diffusion distance instead. And once you have a model like this, you can note that it's a linear model. That's nice because we like linear models. You can use it for a couple of different things. So one is to synthesize data. So if you plug in source densities and all these constants, you'll get pictures looking like this, which is synthetic, which looks sort of similar to the real observations that we have. Of course, it's very hard to tell for a human if a diffuse dot was created by model A or B. But we believe that it's a decent model for these type of experiments. So in this case, we don't have one of these big producers in our synthetic data. But more importantly for this problem is that you can use this as a workable observation model to define inverse problems. And if you actually want to solve anything numerically, you have to discretize. So in the papers that we have written, we develop optimization algorithms over function spaces. But in order to make the presentation slightly more easily digestible, I'll talk about it in a discretized form. So we have a natural discretization in the spatial dimension, which is given by the camera sensor, which is a digital sensor, of course. So the pixels of that camera defines the spatial discretization. Then we have to discretize the movement distance, and that's essentially a design parameter that you can choose to balance complexity versus accuracy of the model. If you're into function space optimization, what we do is what's called an inner approximation of this with step constants functions. Essentially, we simply model the continuous time functions with step functions, and we integrate in order to get the corresponding discrete quantity. If you're a little bit clever about the normalizations, when you move from continuous space to discrete space, you can actually make the norms of the operators involved as well. And this is not important for this presentation, but if you want to do multi-resolution solutions, it's important to keep the norms, because then you can simply jump from one explanation to another explanation at a different discretization. And it's a little bit easier to understand what's going on. And typical sizes of these problems. So if you have sliced the sigma dimension in 9 slices, which we did in a lot of the experiments, and you had a camera sensor, which is 2048 by 2048 pixels, which corresponds to a camera that was actually used, you have somewhere around 40 million variables to optimize over in one of these experiments. And you can consider different kernel approximations in the discrete kernels in order to make it slightly more efficient for computational reasons. I'll come back to that in a little bit. So again, you have the observation model, which is a linear model, and you have the corresponding discrete one. And once you discretize, you can simply treat it as a sum of convolutions of some quantities. So the A here can be viewed as a tensor, where each slice is an image, and you have slices in the sigma dimension. And in our case, in the examples, we have 9 of those. Right, so it's a sum of Gaussian convolutions. So now, once you post it like this, you can consider the algorithmic solutions. And since it's a linear model, we like least squares problems. So that's the go-to solution. Actually, we also looked at the noise, and it's fairly Gaussian in these cases. So it's a good solution from that point of view as well. So we want to recover the source density rate, or the equivalent quantity A. We know that it's positive, so we can use that, and otherwise it's a least squares problem. Now, of course, if you've been following, there is a problem with observability. So we only observe one single image, so at a single time slot. But the quantity that we're seeking is a 3-dimensional quantity. So there's a problem with observability, or the problem is not well posed. And for a lot of people attending this seminar, I think you already know the answer. The answer was in the title, and the answer is group sparsity. So since we know that the solution should be spatially sparse, we can impose sparsity structure of the spatial dimension in this problem. And since it's temporally continuous, since a cell that starts produces over some period of time, we want to group all explanation at the same spatial location into one of these groups where we enforce sparsity. So either something is continuous over time or it's zero. And the way to do this, it's also well known from our literature. You simply add the regularization term with some lambda times an L2 norm, not the squared L2 norm, but the L2 norm for the group. This is how we impose the group sparsity, and this is standard in the literature in the signal processing field. You can also do the same thing in the continuous space. So I will not show the algorithms there, but I wanted to show the expression of the optimization problem. So in this case, we simply moved up the positivity constraint as an indicator function, which is what you do before you apply solutions to solve these problems. Now, I said this, it's still a problem over 40 million variables. So you really don't want to form the matrix A, because that one would be 40 million by 40 million, and try to invert it even, that would be even worse, right? So you really want to work with this as an operator. But luckily for us, both in the mathematics and also in signal processing, there's been a lot of work in matrix-free methods or first-order methods that work with linear operators. And this is actually, you consider the effect of A rather than forming A as a matrix. And also there's been work on proximal operators. So the constraint and the positivity, so the regularization term and the positivity constraint can be handled by proximal operators, which turn out to have closed form solutions, which is very nice. And for the purpose of the project, we have considered a couple of different methods. So accelerated proximal gradient methods, this is what we wrote most about. We also considered multiplicative update rules to solve these problems. So this is actually closer to what's implemented in the actual product I showed you in the beginning. But let's look at the proximal gradient methods. So fairly straightforward once you pose the problem to write down what the Nesterov accelerated proximal gradient algorithm looks for, like this in this problem. So the Nesterov acceleration, which makes it slightly more computationally efficient, also makes it slightly more complex to look at. So let's revert to just the proximal gradient algorithm for a simplified view, right? And once you've done that, all of the terms, they make sense, which is nice. So when you're communicating with someone who is not into convex optimization, you can talk about what the algorithm does nonetheless. So if we go through this, the first step here in line 3 is a model prediction. So we assume that we have a notion of what are the sources that we have in this model. We apply the operator, which is Gaussian filtering, and we get an estimate of what we ought to have seen if this hypothesis was correct. The next step is to compute the residual. So how off were we with this prediction? And that's just subtracting what we actually saw with what we predicted. Then you should calculate the gradient, which in this case is simply applying the adjoint operator on the residual, because it's a least squares problem. And since the essentially Gaussian kernels, or the kernels that we work here, are symmetric, the adjoint operator is more or less the same as the regular operator. So it's not symmetric when it comes to the sum. So instead of having the sum, it simply becomes k convolutions. But the convolutions are the same, both for the joint operator and for the forward operator. So essentially, you convolve the residual with a bunch of Gaussians, and that makes up your gradient step. So what you have inside of the plus here is simply a gradient step, but since everything was supposed to be positive, you project it onto the feasible set. So it's a projected gradient step. And then for any one of you who's looked at the IST algorithm and the FIST algorithm, this should also be somewhat familiar. So due to this regularization, there is a shrinkage term, which forces some of the values down towards 0. And this is the proximal operator that comes out of this together with a positivity constraint. So typically essentially you take the inverse L2 norm across the sigma dimension, and this is the factor which determines how you move towards 0. And then if that happens to become negative, you simply need to set it to be positive again. And then finally, you apply it to the updated variables that you have. And this is the whole algorithm. There are two computational bottlenecks, and that's the convolutions here. So the kernels that you have to use, because the particles can diffuse many pixels in the image, these kernels are quite large. Luckily, they are more or less Gaussian still. They're not exactly Gaussian once you move to the discrete space, but more or less. So what we have here is the sum of Gaussian convolutions of some quantities. And if you look at computer game design, if you want to simulate out of focus areas of the picture, what you do is you do many Gaussian convolutions and sum those up. So this is something that the game developers and the developers of GPUs have worked on for a large time and supplied dedicated hardware for. So if you implement this on a GPU, you can gain a lot of speed in this type of convolutions. And the same goes for the adjoint operator as well. So in order to make this feasible, even though it's a first order method, you really should utilize GPU accelerated hardware in order to solve this efficiently. And you should also try to not treat this as two-dimensional convolutions, but you should try to separate them so you can run them as one-dimensional convolutions. And since we have least squares problems, a linear model, positivity constraints, and an L2 regularizer, everything is convex. So convexity and the theory of convex optimization guarantees that we'll converge towards the global optima, which is very nice. So let me wrap up by some results and the performance of these algorithms. So when you want to look at the performance and compare it to the performance of a human, you need to come up with some metric. So what we essentially do is we compute this L2 norm through the sigma dimension to see how many cytokines were produced by a single spatial location. And this is something that's captured in this norm, which we call the pseudo likelihood. Then we have to set the threshold level. So how large should this have to be in order for it to detect the spot, right? So you have to set it slightly above 0 in order to avoid noise in the optimization. But how much? And the way that we approached this was looking at how people in the business actually analyze this. So typically what you have is you have a sensitivity slider in the software or something that sets the sensitivity. You calibrate that with respect to your test measurement, right? And you find the level that you're happy with, and then you make a comparison between the two. So we look at the number of the precision and recall of detecting the spots in this, and we compute the F1 score. And then essentially we set the levels that yields the highest F1 score. And we can do this both for a human that has labeled these spots and an algorithm which has done that. And that's essentially motivated by this process where you try to optimize the threshold in order to get the best performance out of the algorithm. So that's a bit subjective, but it's fairly robust nonetheless. And you can look, and we have looked at real data and how it compares towards what a human would do. So in this case, the yellow circles that you see in this image are results produced by the algorithm. The orange squares are results produced by a human. So in most cases, it matches very well. Here is a spot that it was detected by the algorithm, and here are two spots which the human thought should be in the data. And that may look a bit suspicious. Why do the algorithm choose this one over these? But then you should know that this threshold here is set on a global level on this picture, right? So it's probably consideration somewhere else to decide how weak can the spots be before they are included. And possibly this spot could also be due to a broken pixel or something like that. But overall, it's a fairly accurate result for this type of pictures. And we can also look at the result of the optimization directly. So in this case, it's for synthetic data. So we have this green picture here, which is original data. And the picture to the right here is the picture that's, or the solution that's generated by the algorithm. And then you can really see that the group sparsity does a very good job of isolating the sources to very localized positions. So there is one error in this particular case, and it's these two spots here, which happen to be simply 1 pixel apart, or with 1 pixel in between them. That's a little bit too narrow for the algorithm, especially since this particular spot is weak as well. But here you also have 2 spots which are nearly 1 pixel apart, which the algorithm successfully breaks. So it is quite good at actually breaking these clusters, which I showed you in the beginning. And if you want to have less anecdotal evidence of how good it is, we did a lot of experiments, a lot with synthetic data, because then we know the ground truth. And we generated this with a much higher fidelity model as well. So it's not the same model that was used in order to analyze the data. And then you can see that in the first one, we look at how does the regularization, what effect does the regularization have? And luckily, it's very insensitive. Otherwise, this is a tuning parameter that you have to set, but it's fairly robust to choices of this parameter. And you see the two black lines here is the performance of the algorithm. The reason that it's two lines is that it's two different way of discretizing the discrete kernels. So one which is using a rank 3 approximation and one which is using a rank 1 approximation. So it truly becomes separable as two 1D convolutions. Then we look at, so this box is kind of interesting. So this is looking at the local maxima of the image, but in a noise-free image. So we feed a local maxima spot finder with the noise-free image. So this would be a Gini-aided denoising method followed by local maxima finding. And you can see that this is significantly worse than what we're able to produce. And if you add noise, you drop even further. And then something which came up when developing this was the question, if you just do a standard deconvolution. So these type of methods have been developed for deconvolution. Could you just apply a deconvolution in order to sharpen the image and then do something? Pick out local maxima, for instance. And the answer is, if you don't regularize the deconvolution, if you regularize it, it starts breaking apart. But almost, but you can never get as much to the same level as a regularized version of the algorithm that we have proposed. We looked at different noise levels, for instance, for the optimal regularizing factor for our method and the deconvolving method. 1, 2, 3, 4 doesn't say that much, but essentially it goes from 10 bits to 4 bits of equivalent noise, additive noise on the models. And you can see that all the methods break down when you increase the amount of noise, but the method that we have proposed is fairly robust to additional noise. And it performs best in all cases, which is nice to know. And then in the final one, we look at the density of spots, right? So if you have more spots, you have more cases of occlusion. And you can see that there is an increasing benefit to the algorithm once you increase the density of spots. So in the last example, we have 1,250 spots, actually in an area which is 1,024 by 1,024 pixels. It's only 1/4 of the real data images. And in this case, the dashed line is quite interesting as well. So one person at this company was trained on synthetic data in order to mark the spots for a couple of images. And we looked at the performance of a human taking a lot more into consideration. So we developed a software where we could set the brightness, for instance. So you can increase the brightness to see the weak spots and so on, and mark them. So the algorithm does slightly better than a human at this task as well, which is nice to know. So the human would be the golden standard in this case, but obviously counting over thousands of spots in an image is not something that you want to do 96 times for one of these wells. So I'm running out of time, so let me just wrap up and related to what's going on and the reason that we're meeting over Zoom today. So this is technology that is already being used. I took a picture from a YouTube video put out by an institute called La Jolla in California. They are currently looking at understanding the COVID-19 virus in order to help vaccine developers with vaccine development. And what they do essentially is they break the virus down to the individual parts of the virus. Then they have a lot of these plates where they add the parts of the virus and they look at how does various parts of the immune response act with respect to these virus particles. And I like this picture because I recognize it to be the software that we helped, we didn't develop the software, but the algorithm or the software where our algorithm was inserted into. So already now, a couple of years later from when we started this, it's being actively used in vaccine development. And then as a second example, this made me very happy. So bringing the loop back to the first presentation of this seminar series. So the first presentation was given by Matthew McKay. He talked about how he used signal processing and computational methods to figure out what parts of the virus are susceptible or good potential vaccine targets. This is also in research that has an impact. So the company that we worked with developing these methods picked up on this paper quite early, and just a few months later, they actually had products out. So you can buy these plates pre-coated with the correct antibodies to test these vaccine targets. And if you do that, you would obviously need access to both the hardware and the software that I've been talking about today. So that ends everything. So summarizing. So what we did was to develop an image processing solution for immunology. All right, here, ELISpot. I only talked about Fluorospot. ELISpot is an earlier methodology using visual light. So we formulated an overparameterized linear model, and then we applied group sparsity in order to come up with a unique solution to this problem. And I also talked a little bit about the role of GPU acceleration in solving these type of optimization problems. So all of this has contributed to a state-of-the-art solution in this particular field. So this piece of software that you can use to analyze these now is really the best you can get for this particular application. In particular, it beats anything that's on the market in terms of breaking clusters of spots. So you can work with much higher concentrations than you can with any of the other methodologies. It has unparalleled positioning of the spot sensors. I haven't talked too much about this multiplexing assay aspect, but in that case, accurately estimating the spots is very important. And then a new thing, because we actually have a model for it, we can make a precise prediction of how many cytokines were actually produced by every single cell. And this is something called relative spot volume now in the business, which is a new measurement that people are just starting to look into what biological relevance this could have. The reason why it's relative is that it's very hard to calibrate for things like exposure and for sensitivity and so on. But in an assay or among assays, the relative metric is very accurate. And if you're interested in reading up on the details, I would point you to the 2 papers in Transactions of Signal Processing that outline this whole thing. So the first part is devoted to the modeling parts, and the second one is devoted to developing the proximal operators. So there is quite a lot of detail when doing this in function spaces. And the second part also contains information about the performance and the performance evaluations, how they are done. And then if you want a popular scientific description, I would point you to the product webpage of this company, Mabtech, where there is a short video presenting similar things which I presented today, but without the math. And they also have a lot of illustrations of how this works. So that wraps up what I had to say. I guess 2 minutes over time. I'll hand it over to the hosts.
Okay, so thank you, Joakim, for the for the very nice talk. So we got a couple of questions already popping up in the chat box. And actually, the first one comes from me.
All right.
So actually from the modeling, so from the sessions when you do the modelings, so I saw that you propose the linearized observation models, and there's a sort of like an A matrix that is like coming from the PDE equations.
Yes.
And that actually depends on something like the diffusion constant or etc. And is it actually necessary for us to fine tune those Lorentz parameters, or do we assume that they are already known to us?
Right. So that's a good thing. So the A matrix here doesn't. So it only depends on assay by setting a set of diffusion distances. There is nothing more in here which depends on the diffusion constant. So so when you move from the source, so the rate at which the cells put out cytokines into the solution until you get to the quantity A here, right? That depends on the diffusion constant. But the step from A to the observation does not depend on the diffusion constant, explicitly at least. You don't need to know the diffusion constant to construct the A matrix, which is very nice because it makes it very generic. If you actually try to obtain like a non-relative measurement of how many cytokines were put out into the solution, then you would have to know the diffusion constant. But typically this is of secondary importance.
I see. So actually that also answers my second questions of whether that is beneficial to also learn the diffusion constant per se. Okay.
Right. I mean, so I don't know. So I don't know the biology, but I would assume that if you could actually not only get the amount of cytokines put out, but exactly when they are put out, right? So when are cells activated during incubation and for how long are they active? I would assume that that has biological relevance. I mean, exactly what relevance that would be, I don't know. But it's also quite hard because it's already an ill-posed problem to begin with that we help with sparsity. So one should be a little bit careful about the reliability of what you actually get out in terms of the source density rate. So we have tried some experiments with that, and it's a little bit hard to get an exact recovery of the actual source density rate. So the spatial locations are easier and the total amount is easier to get.
Okay, yeah. So thanks for answering my questions. So we got the next question coming from Nicholas Gillis. So Nicholas, do you want to ask the question yourself, or do you want me to repeat it for you?
Yeah, I can ask it myself.
Okay, yeah, please do.
So yeah, I'm solving non-negative least squares a lot because I work a lot on non-negative matrix factorization. And you mentioned in the talk that actually in your code you use multiplicative updates. Is that right? Or could you comment on that? Because in practice, my experience says that it's typically much slower than what you presented, so the accelerated proximal methods.
Yes, so it's you're completely right. It's a matter of how. So when you're working as a biologist, how long do you want to wait for the solution? And typically, the tolerance at waiting long for the solution is not that high in the community that this is aimed at. So typically, we have to solve this problem within a second. And multiplicative updates are better in the first couple of iterations. So when you actually try to solve it towards actual optimality of the optimization problem, the accelerated proximal gradient methods are better. Their theoretical convergence speed is better, and they also perform better in practice when you really want to squeeze the last drops out of the optimization problem. But for the first couple of steps, we've found that the multiplicative updates are better. And that's actually quite nice. So we haven't really followed up with that in writing, but there are, of course, so there are well-known multiplicative update rules for positivity constrained quadratic problems, but I have seen coming out of the matrix factorization literature people that have addressed similar regularization as the group sparsity here to incorporate that into the multiplicative updates, and we tested a little bit with that. So I hope that answered the question.
Okay, thanks. So there's just a follow-up question. So you don't really need a high accuracy solution. So typically, do you observe I mean, in the detection part, a big difference if you wait for, let's say, a long time or not. So maybe that's the reason you don't need high accuracy or something.
Yeah. So I'm not sure how much detail to go into, but essentially what is done in practice is so the spatial locations become apparent quite quickly, and those you can get with multiplicative updates. And actually we have a refining step. Since we want accurate estimates of the size of each spot as well, what we do is we run the algorithm for a bit. We identify the spatial locations, then we lock down, and then we have a constrained optimization problem that it's only allowed to explain the data with those spatial locations, and re-optimize in order to get more accurate description of the data with that.
Okay, thank you very much. Okay, so the third question is from CQ Su. So he wanted to know that so by the discretizations, he thought that the transformation A might be regarded as a third order tensor. So here, like the group sparsity can be expressed as the tensor sparsity. Is that a right interpretation of the results?
Ooh, I think I have to show my limited knowledge of a proper tensor terminology. So I would guess yes, but I can't say for sure. Right, so it is sparsity across the third tensor dimension, that is clear. Or groups along the third dimension. So if that is called tensor sparsity, yes, for sure.
All right, does that answer your question, CQ? Okay, yeah, I think he says so. So the next question is from Zhonghui. So please unmute yourself.
Hi, Joakim, thanks for a nice talk. Yeah, I have a very simple question. You showed that the parameter lambda is kind of robust against the noise level. So I wonder how about other factors such as the true sparsity level of or some problem dimensions? I'm curious.
Let's see the. Yeah. Let's see if I understand. So so there is not a notion of robustness with problem dimension, I would say. So so if you increase the number of sigma levels, you have a more ill-defined problem, so to say. But given the group sparsity, and since we're mainly interested in norms or sums over the last dimension, it doesn't really matter how many. So it matters to accuracy if you have more levels, but it doesn't make the problem worse at doing what it's supposed to do. It takes more time if you have more sigma dimensions. So the complexity grows linearly with the number of slices in the sigma dimension. The size of the images, it also grows linearly with the number of pixels because we treat the 2D convolutions. So we essentially decompose them into 1D convolutions. So, and those are only dependent on, oh, let's see, so I'm not dying. No, it grows linearly with the number of pixels in the image. And since so the solution is really local to the image, right? So the good thing of treating the whole image in one run is that you don't get boundary effects. So you don't get these blocking effects if you try to divide it into smaller blocks and do the processing there. You don't really have to do this, because if you have to process all of the blocks and everything grows linear with the number of pixels, that's the scaling that you would have even in a blocking approach. So that is good. And so the image size doesn't really affect performance. Other than the fact that if you have too many pixels, it grows linearly with those number of pixels. There is a bit of a development effort to figure out exactly what the optimal slicing in the sigma dimension is. Typically, you want more slices for low sigma. So details which are already very diffused, you don't need to be so accurate with. Whereas details that are less diffused need more accuracy. So it's a good idea to do a nonlinear slicing of the sigma dimension. I don't really have a notion of robustness to those choices, right? But clearly some of them are slightly better than others. And that's also something that has been investigated in developing the actual software to find out a slicing in the sigma dimension, which works well across all possible type of plates that one could consider.
Thank you.
Okay, so we also have another question from Liu Yafeng. So he is asking, so based on my knowledge, when we do least square and when we also have a non-negative constraints, so in those cases, even if without the regularization, the solution tends to be sparse in those cases. And
Yes.
And the reason is that this non-negativity somehow implicitly promotes the sparsity in your solution. And would you mind commenting on this for the group sparse case?
Yes, I'd love to. This is it's very much true, and it's such a good question that if you want to contact me later to discuss this, I'd be very happy to. So yes, that is true, and you also see that in the images. So even if you don't apply the group sparsity, it works quite well actually, and it definitely has to do with the positivity constraint. And we've seen several examples in the signal processing literature of where this is the case. There are a couple of things that you can observe directly. So one is that since each element in A, so A represents the green function, so the diffusion or the Gaussian. So the capital A has non-negative elements. And that means that the null space of A, which exists, don't run through the positive orthant. So it runs, it only cuts the positive orthant at the origin. And that definitely has something to do with sparsity. So I think if you actually created observation here using this discretized model, I think you are quite close to proving that there is a sparse solution which solves it. I mean, there definitely is a sparse solution. I think it's the only one. It's not as easy as that, but so the columns of A are very collinear still, because diffusion at different diffusion values makes very collinear vectors. So you cannot impose like an RIP conditions, which you typically have in sparse recovery. But the fact that they are from Gaussian distributions means that they have a certain structure. And I've started looking into that structure and partially prove that, so I can prove if I have a single spot, the algorithm will obtain that one as the sparse solution accurately. If you have multiple spots that interact, it is, so I haven't solved that problem yet, but I think there is a solution within grasp to that problem. So it's a very, very good question. It's probably yes, but as far as I know, not solved yet. So it's a relevant question to try to solve as well.
Okay, so thank you. So, okay. So there's a question about whether there are some references for general first order matrix V optimization methods, but I guess those can be found in the references you cite in the paper, right?
Yes, I would typically start with the paper by Boyd on proximal methods and finding reference to the math literature from there. So he is not really focusing on the function space optimizations. The reason why we went into function space optimization was the work of Rade Bott from Austria, which gave guest lectures at KTH. So I guess in his writings, I know he's written a book about this. You can find the relevant references for that, that particular aspect of it.
Okay, so next up we have a question from Matthew, but does Matthew want, so Matthew, do you want to ask the questions yourself? Or should I repeat this for you?
Either way, I can ask it. Thank you, Joakim, for really nice talk and really interesting technology. So I've just got a very simple question about practical application. So the people that are running these sort of immunoassays, what is the major bottleneck? Is this technology sort of trying to replace them, or is it trying to make it more efficient for them, or give them like an initial guess that they can optimize? Or what? So how does that sort of connect with what people are doing in existing?
So I don't know for sure. I've been asking the company as well the applicability of this in those kind of applications. As far as I understand, so it's so you have other assays called ELISA, which is essentially measuring the total amount of cytokines, but not the local locality of them. And as far as I know, those are in use in testing now, or similar technologies as well. So this one is obviously more accurate, right? So if you did this for testing, you would have more accurate responses. But it's also much more expensive to produce these type of assays, right? So they are probably not viable in for testing, but they're definitely viable for vaccine development. And I know that companies that do vaccine development are already using this technology. So I would say it's a refinement of similar technology as ELISA, which is used. So I hope that's correct, but that's my understanding of it.
Right, but I guess in principle, if someone was like looking at these images, right, they could sit there, and if they knew what they were doing, they could try to label everything and do it. It'd be pretty painstaking.
Right. I mean, it's a quantity. So you count the number of spots, right? You have so if you're interested in another protein, for instance, then you have a multiplex assay where you have antibodies for both proteins, and you image those as different wavelengths, right? So they would be separated. So how much you have of protein A or protein B would be obvious because they come in different images. Then if you were interested in question of if there is a single cell producing protein A and some other cell producing protein B, or if there's one and the same cell in the immune response that produce both of them, that you can answer by doing this analysis and then aligning the images.
Right, okay. Thank you.
Okay, so next up we have a question from Gan Law. So are you here? Like, do you want to ask the question yourself, or should I repeat this for you? Okay, so I assume that I have to ask the question on his behalf. So so he said that, so it's very nice that we have an analytical expressions for the observation model, but I guess that the sampling step for the Gaussian basis function may affect the accuracies of the results. And how is the sampling step decided in your experiments? And how does that affect your computation time in this regard?
Right. So it's trial and error, I would say, mostly. So it's there are two decisions. One is to decide on the size of discretization steps in the sigma dimension, right? And that gives different kernels. And then if you look at this picture again with the two top black curves, so when you work in the continuous space and you have a true Gaussian convolutions are separable as a product of 1D convolutions, which is very nice because that's makes it very efficient. When you take a Gaussian convolution and you integrate it over several sigma values, you get a combined kernel which is not separable, at least precisely. because it's many different Gaussians. And when you sum or when you integrate many different Gaussians, it becomes non-separable. But if you integrate over a sufficiently small region, it's close to separable. So what we do in these experiments is that we simply do a rank 1 approximation of the 2D kernel, which makes it separable. You just do that by an SVD of the 2D Gaussian kernel. Yeah, and then the top line that you actually get a little bit extra performance if you do a more accurate one. So then we do an SVD and keep the 3 largest eigenvalue, and then you can do it the convolution as 3 consecutive separable convolutions. So 3 here is 3 times as complex as 1, and you get the small benefit, right? But if you just work with Gaussian, so if you just sample a Gaussian, you're already pretty close, and it's small improvements beyond that in practice. And then the how to discretize the Gaussian in space, that's given by the resolution of the camera, right? And then so the cameras that are used now is 2000 pixels, cover one of these wells. In those, a single spot is around 50 to 100, 200 pixels. So the kernels are of that size, 50, 100, 200 pixels. So they're quite large, but the resolution of the sensor is more than you need, essentially, for the application. You could do, well, not as well, but you could do something in the neighborhood of as well with a lower resolution sensor.
Okay, so does that answer your question, Gan? Okay, so I assume so. So the next question is from Chandra, and would you mind to speak up? Hello.
Yeah, hi, Joakim, can you hear me?
I can hear you.
Thanks for the very nice presentation. This is a completely new area for me, so I have a very sort of maybe a very basic question. You know, these molecules that bind to, you worked out the distribution of the sojourn time of these molecules, but as time progresses, there will be fewer molecules left to which these molecules can bind.
Yes.
And so possibly the sojourn time will increase later in the process. Does your model account for this? I don't know if I'm asking my question very clearly.
No, I think you are. So no, it's not. So that is the, I would say, the major limitation. So in order to actually be able to solve the optimization problems, we really need them to be linear. And if you're working in the low concentration regime, it is approximately linear. So, but once you start to saturate the antibodies, you enter into nonlinear effects, right? Because you can only have as many particles captured as you have antibodies. So there is an upper limit, which is not consistent with the linear model. We've found that, so actually saturating the antibodies is not an issue. So in the fluorospot experiments, even if you have a lot of the cells, you're still working in the linear regime. With the type of assays that we're looking at. This other technique, which is called ELISpot, which works the same in the first step, but then you have a staining which leaves black stains on a white substrate, and you image them with visual light. That black staining tends to saturate. So in the imaging process, you don't have as large dynamic range. So there is nonlinearities that come into the imaging part of the experiment, which is more limiting. What we're doing there is essentially trying to linearize the model as best as we can, similar to, so if you're familiar with this Anscombe transformation that you use if you have Poisson noise but really want Gaussian noise. So we do similar steps for that particular assay. For the fluorospot that I presented today, they are so linear that we don't have to do any deviations from the model in order to handle those.
Yeah, thank you. Understood.
Okay, so just now we have a question from Yandro. So are you here like to ask the questions, or should I repeat it for you? So I guess I'll repeat the question for you. So he got a questions on, so he see that the matrix A is obtained by discretization, so gridding of over the spatial space. So first question is that how do we choose the grid size? And that leverages the resolutions and the problem size. And second, is it possible to apply those so-called gridless techniques in terms of super resolutions to tackle this problem?
Right. Yes. the size of A is so you have typically an image sensor. Of course, since the machine has been built as well, following this project, that was a design variable as well, but it's a fairly constrained. So you have the number of pixels that you have essentially, and they have a certain size. So that sets the spatial discretization. Then in software, you can decide the discretization of the sigma max. But the important thing to know is that there are many different discretization at hand here, right? So you discretized observations, but you also discretize this lowercase a, which is the explanation. And you could discretize that lowercase a at a finer resolution than the imaging sensor. And that would actually give you a larger A matrix or more fat A matrix and super resolution in terms of the positions that you could use to explain the data. And that is something that we have tried in the particular application. So as I said, the spots are at minimum 50 pixels wide across the sensor. So trying to get more super resolution than that is not worthwhile. You don't benefit by, so essentially in a spot of 50 pixels, 1 pixel accuracy is good enough. You don't need sub-pixel accuracy there. But the question, if you move the other direction, actually do coarser quantizations, you would have smaller optimization problems. So it's quite feasible to discretize at the coarser level, do a warm start of the optimizations. You optimize at the coarser level, and then you refine the discretization as you go along. And that's a good idea to do. Then it's so this is something I went through very quickly, but the choice of how you discretize. So if you're a little bit clever, you can essentially maintain the norms of the corresponding continuous space operators there. And then if you do that, it becomes very easy, because then you can essentially just take one discretization bin and split it in two if you want two of them. And the constants, the regularization constants and the step lengths and all of that can remain the same, and you just continue optimizing. So essentially you build optimization problem having the continuous space optimization model as a backdrop, and you think that you're optimizing that really, but at different coarseness in the solutions.
I see. So does that answer your question? So I guess so. So there are no further questions in the chat box currently. So let me ask this as a final call. So is there any further question for the speaker? So if not, so let's all thank the speaker, and thanks for the really nice talk, and also staying behind with answering this Q&A.
Yeah, thanks to everyone who showed up as well, and for all the questions.
Yeah, absolutely.